Yes. Okay, done. So inulin is a polymer of fructose. Can't see the screen. Okay, wait. Okay, just 2 minutes. Yes, Tusha. Can you see the screen now? Is a polymer of fructose. It is a storage polysaccharide of. It is storage polysaccharides of root, tubers. Very important to note. I'm writing the note here above. Inulin is not metabolized in human body. Intent and idea. You cannot see the PP. It's full blank. Just 2 minutes. Okay. Hello. Yes. Yes. Who is this? Ma'am, I'm a little busy. Call me later. Okay, wait. Have patience. I'm trying to connect to a different network. Let me check with that. Ma'am, what's happening to the internet? No, from morning I'm taking class. From morning there's issue. Okay, ma'am, come here. Go take it. Yeah. This right? Level 1 I have WhatsApp team. So it's like photo लेके जाओ. मैं कोई नहीं आया तब से. Level 1 से so many questions. Okay, fine. So tell me all together how many questions done. Okay. Okay, count करके मुझे बताना. I have to give. It should be around 90 questions. Okay. And then when you're making it, one PDF will be the highlight, so showing the answer. The other PDF will be the same thing you go through it. Okay. You just let me know how many questions are. Okay. This level 1 I have emailed. Yeah, what's up to you? On the class. Now can you see the screen? No. Internet issue. Now. Now can you see? No? Dipansha says yes. What about others? Okay, please write down. Inulin is a polymer of fructose. It is a storage polysaccharides of roots and tubers. And inulin is not metabolized in human. Note is a part of inulin. Okay, so inulin is not metabolized in human body. Come on, write down. Do let me know once you're done. Done. Okay, good. Next. Next. Discard. Insert. One more new slide. Come on. Write down about agar agar. Is also a polysaccharide, but it's a type of mucopolysaccharide. It's type of mucopolysaccharide. Okay, and is obtained from red algae, and it is obtained from red algae. It is used as a culture medium in the laboratory. Okay, wait. Can you see it now? Can you see it now? I cannot see any screen there. Okay, one by one. Okay, I think now you can see. Can see now. Next is hyaluronic acid. It is a heteropolymer composed of D-glucuronic acid and DN-acetyl glucosamine. This one. Push. Hyaluronic acid accounts for please write down this. Yes, last word is flexibility. Hyaluronic acid accounts for toughness and flexibility of cartilage and tendon. Account for toughness. Ramachandran. Toughness, tough flexibility of cartilage and tendon. Next. Next. So this is about carbohydrate. We are done with the carbohydrate. Next, we are moving to a second part, that is protein. Let's study about the protein. So can you see the slide of protein, everyone? Can you see the slide of protein? Yes. These are the essential elements in protein are carbon, hydrogen, oxygen and nitrogen. After water, protein is the most abundant component in the protoplasm. After water, protein is the most abundant component in the protoplasm. Proteins are the polymer of amino acid. We know they are the polymer of amino acids. Proteins are heteropolymers. There cannot be homopolymers. They are only heteropolymers. There are around only 20 standard amino acids. Collagen is the most abundant animal protein and Rubisco is the most abundant plant proteins. All are important MCQ questions. So, how many amino acids? 20 standards. Out of 20 standards, how many proteins can you make? You can make infinite proteins. Like out of letters, you can make so many names, infinite names. So, same way, the amino acids are 20 and you can make infinite proteins by the different combination. And proteins are always heteropolymers. Getting the point? Please write down. Done? Next. Now, we all know that proteins are made up of amino acids. So, what are amino acids? Are very important. Amino acids are the group of organic compounds having two functional groups. Important. Which are the two functional group? One is amine group, other is carboxylic group. One is amino group, other is carboxylic group. These are the two functional groups. Amine groups is a basic, whereas carboxylic group is acidic. And then R can be this only for all the 20 amino acid, the change happens only in this R region. So R can be H. If it is H, it becomes glycine. If R is taken by CH3, it becomes alanine. If it is by hydroxymethyl, it becomes serine. So anything that changes in the amino acid should happen only in the R. Rest everything remains the same. One side is amino group, other is carboxylic group. All amino acid contains carbon, hydrogen, oxygen and nitrogen. But some of them will contain additional, those are called sulfur. Physical and chemical property of amino acid are due to amino, carboxylic and functional group present. Amino acids are differentiated into 7 groups. We'll study further. Note, only R changes. Rest everything remains the same. Only R changes in different amino acid. If R is replaced with H, if R is replaced with CH3, then you get different amino acids. Please write down this. Done? Is it done? Yes. Okay, next. Can you see this? This is the general structure of alpha amino acid, and this carbon is called alpha carbon. Here, this is an amine group. This is a carboxylic group. The changes happen in the R. You have written all this. Okay. Now, classification of amino acids. Amino acids are classified further into neutral amino acid, acidic amino acid, basic, sulfur containing, alcoholic, aromatic, and heterocyclic amino acid. What is neutral amino acid? When an amino acid containing one amino and one carboxylic group. Examples are very, very important. One very important slide. Which this one? Okay, please write down. Can you see the previous slide, Komal? Done? Do let me know once done. Yeah. No? Can you see it now? The general structure of alpha amino acid slide, can you see? Yes. Okay, write down.
Ma'am, previous slide.
Previous slide. Amino acid slide. Are you talking about amino acid slide?
Yes, ma'am.
Can you see it now?
No.
No? Now?
Yes.
Okay. Write down.
Thank.
Done? You're done?
Yes.
Oh. Now, amino acid slide. Here, amino acid are classified on the nature. One is neutral amino acid, which is amino acid with one amine group and one carboxylic group. You need to remember the examples, which is a very, very important, and this slide is important. This year also there was a question on this slide. Very important slide. Most of the times the questions will be from here. Acidic is where you have extra carboxylic group, then the amine group. Basic is where you have extra amino group, then the carboxylic group. Sulfur containing amino acid. The amino acid, when they have sulfur, they are said to be sulfur containing amino acid. And this sulfur containing amino acid, examples are very important. Cysteine and methionine are the sulfur containing amino acid. They'll give a group of amino acid and they'll tell you to select which among them is a sulfur containing amino acid. Are you all getting the point? Alcoholic amino acid, the amino acid having an alcohol group, OH group. Which among this is an alcoholic amino acid? Important question. Aromatic. Aromatic means here the amino acid having a cyclic structure. Those attached cyclic structure, those are phenylalanine, tryptophan. Important again. Heterocyclic amino acid means you have a nitrogen in between. Like, you have something like this. Everywhere it's carbon, but you have N in the ring structure. When you have like this structure with the amino acid, it is said to be heterocyclic amino acid. Examples are very, very important. Important, 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 important. Very important. Basic, acidic, everything is important. Please, this slide is important. Every year there will be a question from here. So please revise this carefully. Is it done? No? Okay, write down, understand. Done, everyone? Okay. Any doubts? This is a classification of amino acid based on its nature. Now we'll be studying the nutritional classification of amino acids. Okay. Write down nutritional classification of amino acid. Amino acid which cannot be synthesized just a minute, hold on. Can you see it now? As amino acids. There is a network issue today. Just a minute. Okay, let me. Okay. Can you all see the screen? It's called essential amino acid. Amino acid which cannot be synthesized in the body but need to be present in the diet is called essential amino acid. These are 10 for children and 8 for adults. 10 for children need to be present in diet and 8 for adults. Okay, we'll learn it with a trick. First one is phenyl alanine valine tryptophan 3-renin isoleucine methionine histidine arginine leucine and lysine So the trick is private Tim Hall. So you can combine PVT is for private Tim Hall. So these all are essential amino acids that needs to be present in diet. Okay, 10 for children and 8 for adults. This histidine and arginine can be synthesized in the body of adult. Can be synthesized in adults, but not in children. So in children, it is okay. Can you write down now? Suhas, can you see this? Change the battery after that. You change the battery after that. You change the battery that time you got. So how much you got?
This one, 2,503.
Done. Any doubts? Okay. Then the second is amino acids which are synthesized in the body are non-essential amino acid. Amino acids which are synthesized in the body are non-essential amino acid. A non-essential amino acid. So whichever the rest all comes under non-essential amino acid. Is it clear? Is it clear? Science is so bad. Which are not synthesized in adults. Valine, tryptophan, threonine, isoleucine, methionine, leucine, lysine are not synthesized in adults. Komal, did you got the answer? Komal Joshi. Other than histidine and arginine, rest all are not synthesized in the adults. Okay, let's move to the next. Structure of amino protein structure. Okay, let's move to the protein structure. Now, yes, 8 are not synthesized in adults, 10 are not synthesized in child. Clear? In children. Now, structure of proteins. 4 basic level of structures. One is primary, secondary, tertiary, and quaternary. First, we'll discuss about the primary structures. Primary structure, it is a linear specific sequence of amino acid. Like it's linear. It's a sequence of amino acid. Like the necklace, the beaded necklace. So it's kind of a beaded necklace. Are you getting the point? It's linear. The bond that is present is peptide bond. The first amino acid is known as N-terminal amino acid and the last amino acid is known as C-terminal amino acid or carboxylic amino acid. Is it clear? Primary structure is a linear structure. Are you all there? Done with the slide? Did you understand what is primary structure? Screen? Again screen? Chrisley, what happened? Ma'am, your screen again. Chrisley, re-log in. I think most of them can see the screen, right? What is this? Came back, this back, that back. It's blurred. Okay, wait a minute. Is it blurred to everyone? Okay, fine. Write down. Draw the primary structure, understand, and then write it down. Done? Can we move ahead? Next. Secondary structure. Now when this primary linear structure shows the folding, that is called secondary structure. Okay. It can be alpha helix, or it can be beta pleated sheet, or it can be collagen helix. So secondary structure is formed by the folding of linear structure. So alpha helix example can be a keratin. Here alpha helix example is keratin protein present in hair or nail. It's a right-handed coil. It's a right-handed coil. It is right-handed coil. Are you all there? Yes. It's a right-handed coil. Next. Next is beta pleated. Example for the beta pleated is beta keratin, beta keratin present the scale present in of eyes, feathers in birds. Then very important, fibroin, which is a natural silk. It's the beta keratin, beta keratin protein, which is pleated sheet form. Third one is triple helix. Okay, this is triple helix. 3 polypeptide will coil to form triple helix. Example is collagen. Example collagen. Here 3 protein molecules will twist among themselves. Are you getting the point? Is it done? Any doubts? Can we move to the next? Okay. No doubts, right? Primary to secondary. Primary is a linear, secondary is folded. Next, very, very important is tertiary. Tertiary is formed by folding of secondary structure. Tertiary is formed by folding of secondary structure, right? Examples very important: enzymes and myoglobin. Enzymes remains to be active only in tertiary structure. A very important question. It can fold upon itself or assume a complex but a specific form. It can be something which is spherical, rod, anything, but it has to assume a shape. It should be a geometrical shape. So the secondary structure, when it folds by itself, give rise to a tertiary structure, and the example is enzymes and very important, very important examples. No, myoglobin is a structure. Can you hear me now? Today the net is really bad. I'm sorry. I'm trying, but the network is so bad. Can you hear me now, stars? Screen is blank. Oh God. Now, can you see the tertiary? Can you all see the screen? Tertiary structure. Ramachandra, it's clear. Only Suhas cannot see the screen. Suhas, please re-log in and come. Suhas, please re-log in and come. Okay, please write down tertiary structure is enzyme, myoglobin. Example. What is quaternary structure? Here, proteins are said to be quaternary if they have. The excellent example is hemoglobin. Hemoglobin has how many polypeptides? Come on, tell me. Hemoglobin has how many polypeptides? Come on, tell me. Can you all see the screen now? Yes. Yes. So tell me the quaternary structure having how many, as I said, the quaternary structure is a protein said to be quaternary if they have two or more polypeptide chain. The excellent example is hemoglobin. So tell me how many polypeptides are present in hemoglobin? Come on. How many polypeptides are present in hemoglobin? Four. Very nice. So four polypeptides are present in hemoglobin. And myoglobin, it is tertiary structure. You should know the difference. Myoglobin is tertiary and hemoglobin is quaternary. Is it the same function myoglobin and hemoglobin does, or is there any difference in the function? What's the function of myoglobin and what's the function of hemoglobin? What's the function of myoglobin and what is the function of hemoglobin? Come on. I'm asking you the function. Pratyusha, function. Myoglobin helps in oxygen diffusion, good, in muscles, whereas hemoglobin carry oxygen molecule in blood, right? Now, how many oxygen molecules does myoglobin carries and how many oxygen molecule does one hemoglobin carries? Come on, tell. How many oxygen molecules one hemoglobin and how many oxygen molecules one myoglobin carries? Four for hemoglobin and myoglobin. How many? How many myoglobin? Rakhi, tell me how many myoglobin? Yes, four hemoglobin. How many myoglobin? Myoglobin also carries oxygen in muscles. So how many oxygen molecule does myoglobin carries? Very important question. Very good, Rebecca. One oxygen molecule is carried, Chrisley. One oxygen molecule is carried by myoglobin in muscles. Is it point clear? Very important question. Next. Functional classification of enzymes, of proteins. Can you see the functional classification slide? Can you all see the functional classification slide? Yes or no? Okay. Structural protein. Some of the proteins help in the formation of structures. Those are keratin, collagen. Many proteins are part of enzymes. Any enzymes you can see, give pepsin, trypsin, amylase, any enzyme. All enzymes are proteins. Transport, some of the enzymes are help proteins takes part in transport proteins. Those are hemoglobin, myoglobin. Hormonal, some proteins are hormones, insulin, growth hormones. Many proteins are contractile proteins that take part in contraction, that is actin and myosin. Some are storage protein, such as in egg, ova, albumin. Some are related to genetic proteins, which are present in the DNA. Some proteins are in the defense, such as amino. All the immunoglobulins are proteins, and many are receptors for hormones and the virus on the cell surface. So the functional classification of proteins are different because they are present abundantly and they perform a various function. Right? Do you agree with me? Take down this slide. Any doubts? One, Rakhi, one myoglobin is carried by, sorry, one oxygen is carried by myoglobin in muscles. Myoglobin carries oxygen only in muscles. Is it clear, Rakhi? Any doubts to anyone? Is there any doubts to anyone there? Rebecca, any doubts? Pratyusha, any doubts? Chrisley? Is everything clear to all? Everything clear to all? Deepika? Komal? Okay. Okay, fine. Next. Are you done with the writing? Can we move to the next slide? Next. No? Okay, write down. Quickly write down. Next, on the chemical nature and solubility. Some are simple protein, some are conjugated protein, some are derived protein. Simple protein are compound only amino acids. They are composed of only amino acid residues. Conjugated is you have amino acid along with the amino acid or a protein group. There is a non-protein group. Okay, and derived proteins are those from you get denatured or degraded parts of conjugate and simple protein. Simple protein means it's only made up of amino acid. Conjugate means along with the amino acid you are getting protein part attached, non-protein and protein part. Or derived is something you are getting from degradation of both simple and conjugated. For example, for the conjugate protein like glycoprotein, you have a glyco, which is a carbohydrate part, and you have a protein. So it's a glycoprotein. So that's a conjugate protein. Getting the point? In digestion and absorption chapter, we studied one conjugate protein. Can anybody tell me? In digestion and absorption chapter, we studied one of the, in absorption part, I'm giving you the clue. In absorption part, we studied one conjugate protein. Who will answer this question? Polypeptide. Polypeptide is a protein, right? I'm talking about conjugate protein where you have protein attached to non-protein group. A protein attached to non-protein group. And this we have studied in absorption part of digestion. There is something which is attached to protein and there's a combination of protein and non-protein group. Do you want me to answer? I'll give you a clue. Very good, Jared. Very nice. It's a chylomicron. Remember chylomicron? Very nice. Remember chylomicron? What is a chylomicron combination? Protein plus what? Fat, lipid. Do you remember? So it is a lipoprotein. So it's a chylomicron is a conjugate protein. Are you getting the point, everyone? Yes. Okay. Any doubts? Any doubts? Okay. Next, you can write down this simple protein, conjugate protein, a chart, and derived. Derived is not so important, but yes, this and this is important. Previous slide, which slide? Classification based on chemical nature of solubility. Which slide, Komal? Okay, write down. Can you see the slide? Okay, are you done? Okay, next. Write down the slide about protein simple and conjugated. Do let me know once it is done. Done? Can we move to the next topic? Can we move to the next topic? Any doubts? Okay. Is it clear to all? Can we move to the next topic? Can't see the screen, ma'am. Okay, done. Okay, now everybody listen. Can't see the screen. Tushar cannot see the screen. Still, please relog in and check. Now, I'll give you 2 minutes to revise about the protein before we move to the lipids. Everybody revise about the protein quickly before we move to the lipids. 2 minutes. Come on. Any doubts? Mm-hmm. Any doubts to anyone there? Can we move to the next topic? Tusha, Pooja, Komal, Pratyusha, Ramachandran, any doubts? No doubts. Okay, let's move to the topic. Let's slip it. Lipids are chief concentrated storage form of energy, forming about 3.5% of cell content. Lipids are organic substances relatively insoluble in water but soluble in organic solvents such as alcohol and ether. Functions of lipid: they are the concentrated fuel reserve of food. They are the reserve for the food, right? Lipids are the constituents of membrane structure and regulate the membrane permeability. You know, they are the members of or the formings of plasma membrane. They serve as a source of fat soluble vitamins. They require for the solubility of vitamins. Lipids are important cellular metabolic regulators. Now it is changed. Can you see it now? Today's network is really bad. Now we can see. Chrisley, can you see it now? No? Okay, let me check. Wait. Which slide can you see now? Which slide can you see now? You can see? Protein slide. Okay. Protein wala. No, lipid wala. Can you see? Lipid slide. Can you see? Lipid. One person reply, just one person. Lipid. Yes or no? No. Now can you see the lipid one? Oh God. I go crazy, I guess. Okay, wait. Wait a minute. Okay. Now can you see lipid? Still a promotion, I can see. Is it still? Okay, now can you see? I think you should be able to see now. Lipids. Go crazy with this. Okay. How much time is left? Let me check out. Okay, 5 more minutes. Okay, we'll start lipids in the next class. Is it okay? Okay, anyway, you can see. Okay, fine. 4 minutes. So you can see the slides. Tell me first. Okay, fine. Please write down. Please write down. So tell me, do lipids have a polymer? Do lipid form a polymer? Lipid do not form a polymer. Komal, lipid do not form a polymer. Is it clear? Do protein form a polymer? Do protein form a polymer? Do protein form a polymer? Answer me quickly. Yes. Do carbohydrate form a polymer? Do carbohydrate form a polymer? Yes. Do nucleic acid form a polymer? Nucleic acid? Yes. So, lipid do not form polymer. Please write down this. We'll end up today's class with this slide.
क्लास है आपकी अभी नहीं है, जस्ट
Done? Is it done? You all left me. Okay. Today network is bad, so please don't mind. Yes, done. Deepika, Chrisley, done. Pooja, is it done? No. Okay, write down, write down. And do let me know once it's done. Can anybody do that? Can anybody take a screenshot and send? I think with Suhas has, yeah, they have sent you. You can just check with them. So there are so many friends helping you out there. Okay, are you all done? So let's wind up the class. It's time up. Okay, see you all in the next class. Bye bye. Take care and keep learning. Welcome.
BSc 25 टिकट है, टाइम है 22 मीन्स 10 बजेगा।